Hello, everyone. Thanks for joining us for our latest webinar series. Before we get started, I do just want to let you know that this is being recorded. So if there's anything that you may have missed or we go through pretty quickly, we will have it uploaded to our website and on our LinkedIn so that you can kind of go back and review this series at your own leisure. So with that, my name is Joseph Rogers. I am the commercial manager of stem cells here at REPROCELL. And today I have the great honor of also moonlighting as your host for this series. For those of you who may not be familiar with ReproCell, we were founded in the early 2000s, so 2003 in Yokohama, Japan. And over the past 23 years, we've really crafted our business model to be uh, really the global go-to for all of your different research or preclinical or clinical needs. Of course, with today's focus being hypoimmune iPSC cells, that is one of our strengths and one that we've really established ourselves as a global leader in. So with that, I'd like to just quickly introduce our speaker for today. It is our ReproCell's very own Dr. Luke Wren. He is a stem cell scientist with us and is our resident expert in gene editing. So with that, I will pass it over to you, Dr. Wren, and you can take it away.
Thank you, Joey, for the introduction. Good afternoon, everyone. I'm Luke Ren, and today I'm excited to share our work on hypoimmune iPS cell engineering and related work using a revolutionary gene editing tool called OpenCRISPR-1. I need to mention that OpenCRISPR-1 is a trademark of Perfluent. In the presentation, I also called OC1. So as we all know, the primary challenge we face in iPS-derived cell therapy is immune rejection. While autologous therapies using a patient's own cells can bypass this, they are incredibly time-consuming and very expensive. Haplotype banks offer another alternative way, but can only apply in low genetic diversity countries like Japan are still facing other problems like MHEX, maybe a potential concern even with matched HL types. So ReproCell solution is to generate allogenic starting material with specific genetic modification that allow these cells to evade immune surveillance, effectively treating hypoimmune cells. Those cells can be further edited using landing pad technology, which I will discuss later. So ReproCell's goal is to offer a single source, one-stop shop solution. We not only for generating clinical relevant iPS cells using our non-integrating mRNA technology, but we also provide HLA class 1, class 2 knockout cells and other derivative cells. When we selecting the editing system, ReproCell had a specific wish list. We needed a system as efficient as Cas9, but with also complicated ownership and high licensing fee associated with a traditional Cas9, as well as smaller size, reagents readily available. As we are all aware, like there are debatable ownership of standard Cas9 between the like two Nobel laureates and the MIT professor. So right after Perfluent published Nature paper in 2025 about OpenCRISPR, immediately we turn our attention to it. Just like we saw first robin in our backyard, we know spring is coming soon. So amazingly, we found OpenCRISPR-1 fit all criteria. It's open AI designed gene editing editor. It's very convenient to use. We can directly use the guideline tailored for the regular Cas9. It's royalty free. We have a secure license from the Perfluent for both research and commercial use. and it's very different from the traditional CRISPR system and does not infringe our previous does not infringe on previous patents. And importantly, is have on-target activity comparable to SP-Cas9, and more importantly is have only 5% off-target activity compared to the regular Cas9. So right after we first licensed the OpenCRISPR-1 from a Portland, we first evaluated for the B2M knockout in our iPS cell line using the plasmid-based OC1. We use TIDE analysis to evaluate the editing efficiency. TIDE is a widely used, cost-effective computational method to quantifying the efficiency. It's basically just to commit a sequencing result to the software. This result for this testing are pretty satisfying after trying several low efficiency Cas proteins like MAD7 and other Cas12 proteins. The analysis revealed that 13 out of 22 clones screened were homozygously knockout for the B2M gene editing. We also made a protein from the OC1, then evaluated it by using the gas from the B2M and the C2TA. Similar to the plasmid from the OC1, the efficiency of a knockout for B2M and C2TA is also can reach 70 to 90% for both guys in the iPS cells. So we continue testing the percentage of the homozygous knockout clones in C2TA elaboration using two C2TA guys. So similarly, we identified 9 out of 17 clones as homozygously of C2TA editing in the iPS cells. So how about in other iPS cell lines? So we tested other 5 different iPS lines. We got the same result using our protein from the OC1 for the C2TA. This time with 2 C2TA guys. We can see we can reach almost 90% of knockout efficiency. How about the other cell lines, non-iPS cell lines? So we tested the OC1 in primary MSCs and MSC derived for iPS, we call it iMSC. Simultaneously for C2TA and B2M, we co-transfect the guide RNA and OC1 same time for both guys. So the result is pretty good. Can reach for both B2M C2T can reach 80-90% of efficiency. So we picked one clone from the first testing with 1 base pair deletion in B2M and 350 base pair in C2TA for expansion and QCs. We confirmed this mutation by sequencing. So we also proved the deletion at protein level by FACS analysis for B2M expression in iPS cells and C2TA in CD43 positive cells, as C2TA is not expressed in iPS cell stage. We also proved them stemness by pluripotency marker staining, namely OCT4, SOX2, 9OG, SSE4. So we can say the stemness is still kept. And we also test the karyotyping. It show a normal karyotype. Actually, this cell line is already online for purchase. So by doing this, we can see all this data are proof of principle that the advantage of the OC1 described above are true. By using OC1, we can make the faster progress. As I mentioned above, we are also using OC1 to create the landing pad cell line to facilitate the cell knock-in conveniently like a universal USB port that plug in and play. As shown in the picture, we first created an AA with 1 landing pad cell line and successfully knock in mCherry gene as one safe harbor site, as shown in the picture. We also knock in large CAR construct like CD19 and so-called don't eat me protein CD47 in the AAVS1 site. We are also testing the expression and of those genes right now. We have planned to make the construct knock in the don't eat me protein like a CD47 in one safe harbor and safety switch like an inducible caspase 9 in another safe harbor site. As we all know, the safety is our mainly concern for cell therapy. So we can shut down this cell, we can kill the cell as needed. So importantly, right now we also work with closely with Nevin Point for the bioinformatics analysis with a focus on the off-targeting and uncle panel report for the clinical cell lines. There's a publication last year on Nature nucleotide acid research about the so-called one-step tagging show at the left side. So when co-transfect with a single-strand DNA from the GA and GT containing donor and the gene of interest together with the BX1 integrase plasmid, people can directly knock in gene into the any specific genomic site with only one transfection. So we test this idea using OpenCRISPR, like we showed at the right side of the picture. We successfully knock in amnion green into the collabo safe harbor site. That CRISPR-Cas9 is a mutated form of CRISPR-Cas9. It is widely used for gene inhibition, activation, epigenetics, and chromatin imaging. Just like the dead Cas, by mutating its nucleus domain, we created dead OC1, or sometimes we call DOC1, and we test in the iPS cells. In our test, dead Cas, dead OC1 successfully silenced the OC OCT4 gene. As we know, OCT4 gene is master gene for maintaining stemness. So this inhibition leading to the significant spontaneous differentiation in iPS cells compared to the wild type control, like only within 5 days. So this differentiation is confirmed by the immune staining by OCT4 gene immune staining. So we can appreciate from the result, the right side, the OCT4 was partially disappeared from the colony in this picture. But for the wild type OC1 clone, the most of the OCT4 expression is still there. So very interestingly, the wild OC1 show very high editing efficiency. You can see there's almost 100% or 90% of knockout efficiency compared to the dead OC1, where there's nothing edited for this dead OC1, which is similar to the publications. So we tried another way to show the versatility of the OC1 to make the FUT8 knockout for the antibody production. FUT8 gene knockout can significantly enhance ADCC by enhanced effector function, increased potency, and a lower dose effect, which is very important for the CAR T cell therapy. So as shown in the picture, in the slide, we first made the FUT8 gene knockout in 293 T cells. We picked 2 clones for the later test. So in order to test this idea in 293 T cells, we first transfect the IgG-Fc and Fc-del into 293 cells using the piggyback transposon to make a stable cell line. From the staining, we can appreciate that both Fc and Fc-del expressed on the surface of 293 T cells. As reported, Fc-del could significantly increase the NK cell sensitivity by enhancing Fc gamma receptor 2A, boosted ADCC and reduced CDC interference. So when in our result, when we incubate the NK cells with 293 cells with the FC, there's a little bit of cell NK cell killing, but compared to the FC-DLE, there's a significantly increase of the NK cell killing with similar to the published data. So in summary, Open CRISPR is a powerful, efficient, and most importantly, is royalty-free alternative to traditional Cas9. It offers enhanced safety through the reduced off-target effect and provides a robust platform for both hypoimmune cell engineering and site-specific gene integration like landing pad technology. So thank you for your attention. I'm happy to take any questions. You can contact us at the email provided on the screen.